Hello, everyone. Welcome to the Exon skipping session. We've got a great lineup today. I'm Liana Orlando, the VP of Research at Cure Duchenne, and I'm just going to walk us through a little introduction into what exon skipping is and how it works, and that will set the stage for all of the rest of the speakers that we're going to see today. So, as most of you know, Duchenne is caused by mutations in the DMD gene, and then that results in a loss of the dystrophin protein in muscles. So your DNA sits in the nucleus of all of the cells in your body, and that's sort of the master copy. And we keep that safe. And then anytime we need to use those instructions, a copy gets made, which is called RNA or mRNA. And that then gets read and made into whatever protein. So if you have a mutation in your DMD gene, that mutation is going to carry over into the RNA copy, and then you won't get the dystrophin protein being made. So when we're talking about exon skipping, we're trying to intervene at that RNA step to work with the mutation you have to sort of restore the dystrophin protein. So generally speaking, what is exon skipping? It's a strategy for restoring dystrophin protein in muscle cells, and it involves bypassing or skipping over a part of the gene, and we'll call that an exon, it's a building block of the gene, to counteract the problems that the mutation causes. And this helps the body create a dystrophin protein, which is not quite as long as the full length. It might be missing some portions in the middle. And so it doesn't alter that master copy of your DMD gene directly, but it works on the RNA. But because you're working with the mutation, it only works for some Duchenne mutations. And generally speaking, if we're talking about skipping exon 51, if you have a deletion in 51, we don't have anything to skip. So it's not that you need to have a deletion in the exon you're skipping. Generally speaking, if you have one that's just before or after, this might work for you. But this, to know for sure, this is something you should be talking to your neurologist about or your genetic counselor, or you can also set up a one-on-one meeting with our CARES team. But I'm going to walk you through a little bit, detail how exon skipping works here. So I'm going to try not to kill everybody with the laser. So these blue boxes are the building blocks of the gene. So there's 79 building blocks or exons in the DMD gene. And I'll draw your attention to the shape of the boxes here because that's drawn intentionally. So the boundaries of each exon have to fit together flush for the sequence or the code that's within each of those to match up exactly. And so then, if the boxes look flushed, the sequence is then what we call in frame, and you get from the beginning to the end without any problem. So what happens in Duchenne muscular dystrophy? So in this case, we're talking about an example of someone having a deletion in exon 50. So in the DNA, you're going to be missing exon 50. So in your RNA, you'd also be missing exon 50. And then you can see, well, exon 49 doesn't match up with 51 the way it's supposed to. There's a gap. And so in that case, we say that the reading frame is disrupted, and so you don't end up with dystrophin protein. So what do we do with exon skipping? We say, okay, let's work with that deletion. So in someone who has a deletion in exon 50, if we also give a drug which would mask or cause you to skip exon 51, now exon 49 matches up with 52 flush, the reading frame is restored, and you would then get a dystrophin protein. And so you would get from 1 to 79 with all of the components except missing 50 and 51. So this is one example. It doesn't work just for single exon deletions. In this case, I'll show you with a 2 exon deletion. If you're lacking 49 and 50, then 48 doesn't match up with 51. But if you give a drug to skip 51, 48 matches up with 52, and then the reading frame is again restored, and you could have a functional dystrophin protein. So this is kind of ingenious, right? It's like a puzzle if you're a puzzle kind of person. And scientists are really smart, and the strategy for exon skipping is really kind of an intelligent approach, but they didn't come up with it on its own. It's informed by observing what happens in nature. So individuals with Becker muscular dystrophy tend to have mutations which, even if they're missing the exons, the reading frame hasn't been disrupted. So we know that even without the full length, as long as you have portions of the dystrophin gene, you might have a slower decline in the disease. It might be a milder form of the disease. And even in individuals with DMD and have out-of-frame mutations, sometimes the body has figured out how to naturally skip on its own. And so if you've been told that you have something called revertant fibers, or that you're in your muscles you have like 1 or 5% dystrophin, that's because your body's probably figured out a way, even with that mutation, to restore the reading frame and still produce some dystrophin. And that is very often associated with a less severe cause of the disease. So, we take what nature has taught us and then say, okay, can we develop some therapies which take something that happens at a low level in some individuals and make it happen at a higher level in more individuals? And that's the basis for the therapeutic approach of exon skipping. So when you work through that puzzle and look at the different exons that you can skip and the prevalence, it turns out that approximately 80% of individuals have genotypes which would be amenable to exon skipping. But when you look at each exon and what proportion of individuals with Duchenne it would work for, you see that it's smaller amounts with, for example, exon 51 being, can work for approximately 13% of individuals with Duchenne. And if you take all of the FDA-approved drugs, we're very fortunate. We have 4 FDA-approved drugs for exon skipping, and they address the first 3 wedges here, exon 51, 45, and 53. That addresses up to about 29% of Duchenne cases. Fortunately, we also have additional programs in clinical trials, and the goals of those additional programs are multiple. We're trying to target additional exons so that more mutations would have therapeutic options. We're also trying to increase the amount of dystrophin which is restored, and not just in skeletal muscle, but also in cardiac muscle. And because these therapies, the FDA-approved ones, they target the RNA, and since the RNA is continually being produced, the drug has to be given regularly. And so the FDA-approved drugs are weekly infusions, but some of the ones you're going to hear about are trying to lengthen the time between treatment and getting out to maybe monthly infusions or longer. So this is a busy slide, but I think it speaks to just how robust an area of research this is. And so we have a great lineup of companies, some of them talking about drugs which are already approved, some talking about clinical trials which are active and ongoing. And I'll tell you, in addition to what's listed on this slide, lots of companies also have other programs addressing other exons sort of in the wings waiting. And once they get, their first program across the finish line, with approval, they're ready to work on the next exons as well. So with that, that's the introduction to exon skipping, and I have them in order here. So the first person we're going to hear from is Wendy from Sarepta Therapeutics. Thanks.
Thank you. Hi, everyone. Thanks for having us. We're super excited to be here. I do want to acknowledge, first of all, Liana did a great job giving an overview of exon skipping, but the last slide she showed makes it very clear to all of us that there's more to do. So while you see a lot of the same, let's say, more common exons represented in research, we acknowledge that that's not enough. So with today's presentation, I'm going to talk about a couple pieces of data that we've recently made public and shared on the Sarepta exon skipping programs. This presentation is meant to provide education and scientific information. As always, if you have specific questions about your needs or your child's care needs, please talk to your physician. So at Sarepta, as Liana mentioned, we have 3 approved exon skipping therapies: Exondys 51, Vyondys 53, and Amondys 45. You can access more information with the QR code here. This is the meeting of QR codes. What's really important to look at is sort of the dates on this slide. So this work and the research behind it and what got these medicines to market has been going on for a very long time. But pretty importantly for everyone in here, and just take a minute and pause and reflect, if we think about today, 2026, what happened in 2016 was the first approval. So that's a minute for everyone to just think about what the Duchenne community has done together. What you see on this slide, if it skips, these are muscle biopsies of patients with Duchenne showing the damage that starts at a really early age. And why this matters is we know time is muscle. But what you see here are muscles of little guys, ages 1, 3, 5, and 8 years old. And it's really hard to impact or show any potential recovery in muscle tissue after disease progression to what's on the far side of the slide, the fibrotic phase. So we know that this happens early, and we know early intervention is probably better. I'm going to shift gears a little bit, and then we'll get into the data. At Sarepta as a company, we're advancing 3 areas of proprietary science. You heard Liana talk about some different ways of hitting where things are happening in the cells. We have an siRNA program, which is a silencing program. We have gene therapy, which you heard about yesterday with the Elevidys, and that adds a functional copy of a transgene of a missing or malfunctioning gene. And today we're going to focus on RNA exon skipping, which Liana just explained. I'm not going to go into this slide in detail, but really wanted to show everybody the breadth and the depth of the research that supports the exon skipping therapies that we've been fortunate enough to bring to market and get across the finish line with FDA approvals. Data are available for all of these PMOs or exon skipping drugs from a wide range of clinical trials and real-world studies. So real-world studies are those that examine the impact of a medication in normal clinical care, so after a phase 1, 2, or 3 study. All of this data really does include ambulatory and non-ambulatory people who range in age from 6 months to 33 years old. This is really important because there's a wealth of data that supports this work and the impact of exon skipping. At Sarepta, we are genuinely committed to transparency and communicating scientific learning with the community. So today I'm going to focus on 2 specific studies under phase 3, Essence and Evolve. Essence is a randomized, double-blind, placebo-controlled phase 3 study. What does that mean? That means that we worked with the community and the FDA a long time ago, 9 years ago when this study was initiated, to evaluate exon skipping therapy in a subset of people with Duchenne in a very rigorous scientific way to prove a hypothesis of what we thought these medicines could do. So we listened to the community and did a clever study design to limit as much as possible the number of placebo patients and not risk the rigor of the science. And with that method, we also, everybody that was in the placebo arm was able to roll over to active drug after a 96-week period. So we'll talk a little bit more about Essence. In this study, as I said, patients were treated with either Vyondys or Amondys, and you can see the regimen here. What's really important to just kind of call out here is the ages were 6 to 13 for exon 53 skipping and ages 7 to 13 for exon 45 skipping. Patients were on a stable dose of corticosteroids for about 24 weeks prior to the first week. And the endpoint, we were talking to a family today about how we need to evolve endpoints. But remember, at the point in time that this was studied, what we knew could be a potential indicator of efficacy and safety was the 6-minute walk test. So that is an endpoint in this study, as well as FVC. So the results. We announced earlier this year and again at the MDA Scientific Congress, and we're very happy to bring this information to the community today. The primary endpoint was 4-step ascend velocity. What does that mean? How fast someone is able to accelerate 4 steps. With that endpoint, the primary endpoint, that the 4-step ascend velocity declined slightly less in golodirsen and kasmirsen, so Vyondys and Amondys, in that group more so than placebo, but the difference was not statistically significant. So we'll talk about that a little bit. Also, in the treatment groups of Vyondys and Amondys, we showed a greater increase from baseline at weeks 48 and 96 compared to placebo. I'm not very good at math. That's a long period of time. And why that's important is it takes a while in a condition like Duchenne to actually show the impact of intervention. So it's a slow-moving, progressive condition. There are a lot of factors. So that is really important to think about how you interpret information and results with studies in Duchenne. These medicines were very well tolerated over 144 weeks, and this is consistent, with what we saw in previous clinical and real-world evidence. So in the post-hoc analysis, what's a post-hoc? That's an analysis that's done by researchers after a study concludes, which aims to really sift through the data in different ways, understand, tease out the important points of the data, and answer potentially other questions that weren't contemplated in the very beginning. So it's a really important part of research. In this post-hoc analysis, there were 2 very big considerations. One is Duchenne is heterogeneous. Chris Curran, what does that mean? Is he in here? We talked about this morning. Heterogeneous means, and for all of you in the community, you know this, there really aren't 2 kids that have the exact same presentation of the condition, even if they have the same mutation. So thinking about how you assess efficacy of a medicine in a population like that, we had to take that into consideration. The second one, and not to give anybody PTSD, but if we go back to this time period, we were all in a world of COVID-19. So we realized as we did the readout from this study all these years later that we needed to stop and think about the impact of COVID. Why does that matter? Families and families with Duchenne, the whole access to mobility and care changed during that period. This was also a very large global study, multiple countries, and COVID looked different in all of those geographies. So we re-looked at the data assessing the impact of COVID. So that was really important. In this post-hoc analysis, the Amondys and Vyondys group showed a reduction in 4-step ascend decline, so better, and they also showed that there was an impact from COVID. No new safety signals were observed in Essence. Vyondys and Amondys are well tolerated. Dystrophin expression increased in both treatment groups versus placebo. While the primary endpoint was not met in Essence, the additional analyses were performed to help better understand the results and what's happened over time. Highlights really do show how the real world impacts clinical trials. This all adds to the body of evidence that supports that golodirsen and kasmirsen, Amondys and Vyondys, may slow disease progression. I also want to just show that across the clinical and real-world setting, evaluation of exon skipping, there's really important information that we've gathered through continuing to invest in research and scientific learning and being able to share that with the community that shows functional impact on the things that are most important to families with Duchenne. Ambulation, respiratory function, cardiac function, survival, patient experience. There's a very high compliance rate with weekly infused exon skipping therapy, which speaks to the patient experience, this commitment to continue to receive medicine. The safety of these drugs has been evaluated, as I said, in a wide range of age ranges, both ambulatory and non-ambulatory patients. And what the data shows is average loss of ambulation in the kiddos that have been treated with exon skipping is 15.8 years old. All right, I'm going to shift gears a little bit and talk about Evolve. Evolve is a phase 4 study. So what does that mean? After you get through the clinical phases of a drug being tested in a scientific academic center, for example, phase 1, 2, 3, phase 4 is an opportunity to continue to collect data outside of that rigorous clinic, so to speak, but more inclusive of extra time and an extension of the original study. So this is an observational study to collect available data from the guys with Duchenne receiving eteplirsen, so Exondys, Vyondys, and Amondys. All of these treated kiddos receive each of the drugs, the actual drugs, so not an investigational medicine at this point, at the time of study enrollment as prescribed by their doctors. The assessments in this study covered those same functional outcomes that I just mentioned: loss of ambulation, time to rise, 10-minute walk run, performance of upper limb mobility or pull. And all of these things are really those functional measures that matter to families. And then from the clinical outcome side, pulmonary function is measured as well as cardiac function. So we don't have time today to dig into those data, but we have those data. So reach out to us. We're happy to talk here or after we all go home. The objective of this study is really to continue to understand the usage, safety, and clinical outcomes of long-term use of exon skipping with these medicines that are now approved. So we did an interim analysis early last year, and of the 173 patients that were enrolled, the majority received Exondys. Why? That study started earlier. It's also the most common mutation. So there's a reason why there's so many more kiddos on Exondys. The percentage of patients staying on therapy, as I mentioned, week in and week out, kudos to the caregivers, really remains high over the course of time. And the overall safety profile is consistent with no increase in side effects or adverse events. I mentioned earlier the median age of loss of ambulation in this new world of exon skipping is 15.8 years. So just from a summary perspective, Evolve continues to support the long-term impact of Exondys, Vyondys, and Amondys, and on clinical outcomes such as time to loss of ambulation. And give everyone in this room a pat on the back again. 10 years ago, the first drug was approved for Duchenne, and that's Exondys. And together as a community, we're really leading the way across rare disease drug development. Reach out to us at any time. We have an incredible program of case managers. I've heard from many of you over the last couple of days how important your case manager is. So Sarepta Assist is our patient support program. They're here to help. They're here to take some of the burden off of you. And you can always reach out to the advocacy team at advocacy@sarepta.com and also Sarepta DMD. But come see us at the Lego table, which is being set up now, so we'd love to see everybody there today at this meeting. Thank you.
Thank you, Wendy. That was terrific. So now we're going to hear from NS Pharma. We have another company with a FDA-approved product. So let me welcome Cassandra to the stage.
Hi, everybody. It's so great to be here with you today. Thank you to Cure Duchenne, as well as the community for allowing NS Pharma to speak. My name is Cassandra Caulfield. I'm a medical science liaison with NS, and today I have the absolute pleasure of telling you a bit about Viltepso. And this slide is just indicating some forward-looking statements that I may make during this presentation. Okay, so what is Viltepso? Viltepso is an antisense oligonucleotide that's indicated for the treatment of Duchenne muscular dystrophy in patients that have a confirmed mutation that is in the DMD gene that is amenable to exon 53 skipping. And this indication has been approved under accelerated approval based on the increase in dystrophin that was seen in skeletal muscle in patients treated with Viltepso. The recommended dosing is 80 milligrams per kilogram of body weight, administered once a week as a 60-minute intravenous infusion. And if a dose is missed, make sure you talk to your healthcare provider to get that next dose as soon as possible. Here's just some safety information. In clinical studies, there's been no reported events of kidney toxicity during treatment with Viltepso. However, in drugs like Viltepso, kidney toxicity can occur, so it's important that you monitor the health of your kidneys prior to the start of treatment and throughout the duration of treatment. Some common side effects include upper respiratory tract infection, injection site reaction, cough, and fever. And I love this slide because it really nicely illustrates NS Pharma's deep commitment to the DMD community. We have Viltepso approved in the US and Japan, but we also have much in our pipeline that we are very excited about. So today I'm going to walk you through 2 clinical trials. I'm not going to throw a bunch of data at you, but there's going to be 3 slides that I'll point out with key information that I want you to take from this. So let's start with clinical trial number 1. It's a phase 2. And here's the clinical trial design. It is a story of 16 boys that were split into 2 groups, 8 of which received a low dose, which is the 40 milligram per kilogram, but the remaining 8 received a high dose, which is the FDA-approved dose of 80 milligrams per kilogram. And this study started and ended with a muscle biopsy, and we also looked at safety and efficacy. So who participated? This was a story of purely ambulatory boys that were between the ages of 4 to right under 10 years old, and they had to be on a stable dose of glucocorticoids for a minimum of 3 months. It was a once-weekly infusion. The duration was for about 25 weeks, of which I'll show you. And we measured dystrophin levels through Western blot. That's just a way to measure protein. And there were some secondary endpoints that I won't get into today. Okay, key slide number 1. We're looking at the mean amount of dystrophin. And this slide right here is showing you the boys that were treated with the high dose, the FDA-approved 80 milligrams per kilogram. And as I said earlier, this study started and ended with a muscle biopsy. So in the gray, you're looking at their baseline, that first muscle biopsy, where the mean normal dystrophin was 0.6%. But after that 25 weeks of a weekly 80 milligram per kilogram dose, these boys had an average of 5.9%. And this was statistically significant. And what I also want you to take away is that 100% of the boys represented in this graph had an increase in dystrophin from baseline. Here is the safety profile from this clinical trial, and what you need to take away was that there were no discontinuations, no deaths, and no serious treatment-related adverse events. Okay, time for clinical trial number 2, and this involved both ambulatory and non-ambulatory boys, and we call this our Galactic 53 trial. Okay, so this is a separate cohort of boys from the first, and this study duration was about 49 weeks, so about a year. And this was 10 ambulatory and 10 non-ambulatory boys. And while we didn't have a placebo, the way that we can compare to controls, we use the natural history study, which I'm sure many of you are familiar with, and we're able to match the boys that are treated to boys from the natural history study on age, ambulation, and steroid status. So who participated? I've already mentioned a bit of who was in this trial, but what you can also take away from this was that now the minimum age was 8 years. It was, again, a once-weekly IV infusion for the duration of about a year. I'll talk about safety and tolerability, oh my goodness. And the key secondary endpoints that I want you to walk away from is that we looked at respiration, so lung function, as well as that performance of the upper body. So how do we look at respiration? How do we test lung function? We use something called forced vital capacity. And what it is measuring that exhalation, that maximum exhalation after a deep inhale. And the way that we measure the performance of the upper body is through a test called the Performance of the Upper Limb 2, or PUL 2.0. And this task, there's multiple tasks in this test that involve such as moving and lifting weights, or measuring the ability to lift one's arms above the head. And what's really cool about this is that you can take individual scores from the shoulder, the mid-level, as well as the distal abilities, but you can also individually look at these to see how they might be changing over time. So let's look at the lung function outcomes. We divided between the ambulatory and non-ambulatory populations, and in the light blue is our ambulatory boys, and in the dark blue bars are the boys that were treated with Viltepso. And the mean change from baseline in the Viltepso boys was 8.3%, while the natural history study was 1.2. And in that non-ambulatory population, the Viltepso-treated boys had a mean change from baseline of 1.6, while the natural history cohort had negative 3.2% change. Okay, let's talk about that performance of the upper limb. Divided again by ambulatory and non-ambulatory participants. And on your left, you can see the total score, the sum of that distal, mid-level, and shoulder score put together. But then we also looked at that individual mid-level elbow score. And why did we do that? This score, as we know, is important for independence, being able to feed oneself, to drink water, to even brush your teeth. And what I want you to take away from this slide is that over the course of a year, these lines are straight. They're stable. Here's that safety profile, which was very similar to our first clinical trial that I talked about. No serious treatment-related adverse events, no discontinuations, and no deaths. Okay, and what I would like to leave you with is our number. So NS Support is a customer support program with dedicated case managers who will help patients, the guardians, and the healthcare providers obtain benefit verification, as well as information about product administration in a hospital outpatient department or a home infusion setting. Lastly, on behalf of NS, thank you so much to all of the patients, their dedicated families, and the healthcare professionals in the DMD community that make these studies possible. I think it was Walt Disney that said Disney is the most magical place on earth, but obviously he hasn't been to Futures. Thank you guys so much for your attention, and enjoy the rest of the conference.
Thank you so much, Cassandra. That was so sweet. I'm going to use that. So next up, we have the first of the presentations for companies which are in active clinical trials. We're going to welcome to the stage Beth from Avidity, which is now a Novartis company.
Thank you. Hello, everyone. Nice to see familiar faces in the crowd. My name is Beth Henriksen, and I am the Senior Director of Patient Advocacy at Avidity Biosciences. Now, some of you may have heard that Avidity was recently acquired by Novartis. So now Avidity is a Novartis company. As such, our presentation will look a bit different than you may be used to seeing in the past. So if you have any questions, please come find our table, and we'll be happy to answer those for you. So today I'm here to share an update with you on our clinical programs, our Explore 44 and Explore 44 OLE clinical trials. These trials are studying an investigational product we like to call Delzota, which is being studied in boys and men amenable to exon 44 skipping. Oh, there's my title slide. Next slide, a couple of disclaimers. The information we're sharing with you today is about an investigational product, which means it hasn't yet been approved by the FDA. So this information today is strictly for educational purposes and is current as of today. So a brief introduction to Avidity and Novartis. I'm sure most have heard of Novartis, right? Raise of hands, anybody heard of Novartis? Yes, okay, that was to be expected. But not everyone may know that Novartis has a presence in neuroscience and has for the past 80 years, and currently has approved therapies in both MS and SMA. And at Novartis, the ambition is to create an impact with innovative medicines that are changing the course of disease progression. How does Avidity fit into that? Well, we've been acquired neuromuscular, so now Novartis is committed to neuromuscular DMD, FSHD, DM1. So today we're here to talk about DMD. Novartis is focused on RNA therapies, genetic therapies, and like Avidity, has a commitment to you all in the room, to the community, to make sure your voices are represented in all the work that's being done. Okay, Avidity, same question. Has anybody heard of Avidity? All right. So with Avidity, we're focused on RNA therapeutics as well, and the delivery to treat rare, tough-to-treat genetic diseases, and looking at the root underlying causes of these diseases. How are we doing that? We have a proprietary technology that's delivering del-zota directly to muscle cell. This is our science. It's a mouthful, but an antibody oligonucleotide conjugate, or we like to call it our AOC. So this AOC platform has a few pieces, and what's rendered for you on the screen is on the left-hand side is our antibody. In the center is our oligo therapy, and those things are what's called conjugated to then form the AOC, which is our drug. So the delivery of that is represented here in this cartoon. So starting at the top left-hand corner, we have the antibody, the oligo, and then it binds to that orange circle, which is something known as a transferrin receptor. And what that does, it is enables to bring del-zota into the muscle cell, allowing those oligos to go even further into the nucleus of the cell. And as Leanna mentioned, we have that reading frame, right? And that's depicted at the bottom of the screen. And it's just a part of the reading frame demonstrated up here. But that oligo then enables that exon skipping that you heard about to take place. And again, with exon skipping, the goal is to increase dystrophin production. All right, so far so good? Okay, great. So I'll share now a little bit about our trials with you and where we are today. We have a phase 1 and phase 2 trial. So phase 1 is depicted on the left, and this trial is now complete. So we had a healthy volunteers cohort, and then also a cohort of 26 boys and men living with Duchenne, amenable to exon 44 skipping. We had a placebo control in the trial, and also had a 5 milligram per kilogram and 10 milligram per kilogram dose. We had boys ages 7 to men up to age 27, both ambulatory and non-ambulatory participants in the trial. Now, focusing our attention to the right-hand side of the screen, this is where we are in present day, which is our Explore 44 OLE trial, which stands for Open Label Extension. So it's a 2-year study studying the long-term safety and tolerability of del-zota. So in this trial, we have 39 participants. Most of, well, not I can say most, but yes, most of them which had rolled over from that phase 1, 23, and then we welcomed additional individuals into the trial there with a count of 16 individuals. Now we made the decision to lock in on a therapeutic dose of 5 mg per kg, so everybody who's on this OLE trial is receiving that 5 mg per kg dose, and that's administered every 6 weeks, so once every 6 weeks through IV infusion. It's hard not to ask if there's any questions, but please do come see us if you have any. Now, with any clinical trial, there's going to be objectives. What are we working to study? What are we working to learn? And with del-zota, our primary objective is anchored on safety. So in phase 1, it's all about safety and repeat dosing of del-zota. The secondary objectives focus on, there's so many big words in science, right? So two things. The pharmacokinetic profile, or the PK profile, which is looking at what the body does with the drug. And then also the pharmacodynamic profile, or the PD profile, which is looking at the drug effect on the body. Well, what does any of that really mean? So the PD profile is looking at things like exon skipping. What percentage of exon skipping is happening? And then importantly, dystrophin production. Is dystrophin production happening when people are receiving del-zota? Exploratory objectives that are also important for us to look at include muscle function and mobility. So tests you're all familiar with, including the PUL tests for our non-ambulatory men and boys. Pulmonary function is also being looked at, in addition to, importantly, patient-reported outcomes. We also have some additional measures we're looking at there on the slide. Okay, now taking us where we are in present day is a CS2 study. Our primary objective, again, is anchored in safety, but this time looking at that long-term safety of del-zota. Our secondary objective now is looking at CK levels, right? We know creatine kinase is a measure of muscle damage, and in Duchenne, you want that number as low as possible. So we're looking at that to see treatment effect. Is there an effect with CK levels when people are receiving Delzota? And then, like phase 1, the exploratory objectives, looking at functional measures, pulmonary, and then also those are patient-reported outcomes. There's a clock down here that's counting down, so I'm trying to race the clock to be mindful of everyone else. But here, there's a lot of data on the slides, or a lot of words, and I apologize for that. But in summary, this slide is intending to let you know that all those objectives and endpoints we just talked about are being met in our trials. So the data is demonstrating things such as a mean increase in dystrophin. What does that mean? We're seeing dystrophin is going up. Also seeing that reduction in CK, so CK is going down compared to baseline over an extended period of time. So this data supports where we are right now in phase 2, but then moving forward with our plans with the FDA to file our BLA, it's called, we'll talk about in the next slide, for under the accelerated approval pathway. All right, so we are aligned with the FDA on our path forward, and Delzota has been granted what's known as fast track designation and breakthrough therapy status, and we're on track to file what's called our biologics license application, our BLA, in the first half of the year. So that's coming soon. And with that, then we're also looking forward, what is next? Now, Avidity has a number of exons in a pipeline, and so next for us is looking at exon 45 skipping for the boys and men who are amenable to 45 skipping. Now, none of this would be possible without you, the community, you sharing your perspectives and your time. So we would like to sincerely thank everyone, particularly the participants in our trial, the families, the PIs, site coordinators, everyone who's really helping us advance this science on behalf of the Duchenne community. Now, we would love to stay in touch with you, so please, if you're so inclined, get out your phone to scan the QR code. You'll be prompted just for your name and email address, and as we have information to share, we can email that to you directly. So that does it for me today. Please do come see our table with any questions, and we'll look forward to connecting with you then. Hope you enjoy the rest of the conference. Thank you.
Thank you so much, Beth. She did a great job introducing us to the transferrin receptor, which is the thing on the outside of the muscle cells that helps pull the oligonucleotide into the cell. And we're going to hear now from another company, which is using that same sort of targeting mechanism. So Lena, please come to the stage and tell us about Dyne Therapeutics.
Thank you, Leanna, and welcome. Good afternoon. I'm Lena Stad. I'm a medical science liaison with Dyne Therapeutics, PharmD by background, pharmacist. And today it's my great pleasure to go over some of our top-line data. And before we get started, I'd like to take the opportunity to be here. Thank you to Cure Duchenne. I also want to acknowledge Cure Duchenne for the early investment that was made in order to make this possible. So thank you for that early investment for Cure Duchenne. So today, like I said, I'm going to be talking about the top-line result with our phase 1/2 trial of zilizumab rosudirsen, or short for Z-rosudirsen, in exon 51 skip amenable DMD. So at Dyne, we're on a mission to deliver functional improvement for individuals and families with Duchenne. And we feel that functional improvement is best served by addressing the root cause of the disease. And I'll go through the mechanism of action with you shortly. So here is the pipeline, and if we look here, you'll see at the top, we have our first target is exon 51, and so this is Z-rosudirsen, and we are planning on a BLA submission for Q2 2026. In addition to exon 51, we also have other exons in our pipeline, 53, 45, 44, and 55. And beside DMD, it's also an exciting time. We do have other neuromuscular disorders in our pipeline as well. That also includes DM1 and Pompe and FSHD. Oops. I think I went. So Leanna did a nice overview of the mechanism of action as far as PMO. This right here just shows you kind of the mechanism of action for zerosudirsen, which is an investigational treatment, using the FORCE platform. So it is a fragmented antibody, so you can see it is a third of the antibody, so you can see just a third of the size. And it has 3 different components. You've got the antibody, the linker, and then the payload. And as I mentioned, we really want to address the root cause of the disease by getting the drug to the targeted tissue. So in this case, the skeletal muscle, diaphragm, cardiac, and CNS. This is an overview of the DELIVER study, which again is the phase 1/2 study with zerosudirsen. It is a 6-month placebo-controlled study with an open label extension. We had ages 4 to 16 years of age, ambulatory and non-ambulatory participants. And the dosing is zerosudirsen or placebo every 4-week infusion. For the primary endpoint, we are looking at change in dystrophin at 6 months, as well as looking at safety and efficacy. And I will also touch on the the select functional endpoints that are listed here. For the cohorts, we had a multiple ascending dose cohort that included 54 participants that were on the from the MAD cohort, we went to the 20 milligram per kilogram every 4-week infusion. And that was the registrational expansion cohort dose, and that included 32 participants. The safety profile for zerosudirsen, there were no treatment. The most common treatment AEs was pyrexia, fever, and headache, and that was on the 20 milligram per kilogram dose every 4 weeks. Oh, thank you. And I just also want to make sure that I highlight that there hasn't been any treatment AEs or related to any study withdrawal, and no treatment AEs leading to death in the study. We did have 2 related serious treatment AEs on the 20 milligram per kilogram every 4-week dosing. That was fever and malaise. And then we, in terms of additional safety data with the 20 milligram per kilogram every 4 weeks, no participants have had any persistent anemia or thrombocytopenia. And to date, we've had over 1,000 doses of zerosudirsen 20 milligram per kilogram every 4-week administered. Now we can go to the next slide, please. Thank you. So here is the primary endpoint. And here on the, I think I'll just leave this. On the blue graph, the blue line is representing the zerosudirsen arm, and the gray is representing the placebo group. And you can see the both adjusted and unadjusted dystrophin level, and you see that there is a 7-fold increase for both values, and that was statistically significant. So I mentioned to you there were other selected functional endpoints that we also looked at. So here we're looking at the increase in time to rise velocity, as well as the 10-meter walk run at 6 months. Okay, so we're going to go back. So this is, we're going to go through each of these selected endpoints that are very important in terms of assessing muscle function in DMD. So you guys are probably familiar with all of these, but just to level set, the time to rise velocity, the higher this value, the faster your ability to stand. The 10-meter walk run, the faster this value, the faster you're able to walk and run, and you have better mobility. The NSAA, the faster or the higher that value, the more better motor function you have. And then the SV95C, the higher this value, the faster you're able to walk and run. The PUL 2.0 is the higher these values are, the more independent you're able to move your arm and hand. And then lastly, the force vital percent predicted value is the higher this value, the better you're able to breathe. So it's a lung function assessment. So the time to rise velocity and 10-meter walk-run velocity, here are the numbers. Just to orient you to the graph, the blue line is representing the zerosudirsen arm. The gray line is representing placebo. And you can see that there is improvement from baseline to 6 months for both of these endpoints. We also saw an increase in NSAA and SV95C at 6 months. So again, the blue line is the zerosudirsen arm. The gray line is the placebo. PUL 2.0, as well as the force vital capacity percent predicted, those numbers were also improved at 6 months. And so we didn't get a chance to go through the 24-month functional improvement, but so we do have, I just summarized all of the 6-month functional improvement versus baseline for these 6 measurements, but we do have data that does show that there is a 24-month functional improvement versus baseline with zerosudirsen for these assessments as well. So now quickly, I just, since we've had some technical issues, I want to make sure that you're also aware that we do have long-term data. And this is kind of looking at, okay, so I just shared with you the 6-month data, but really looking at zerosudirsen and what does it look like comparing it to natural history data. So the blue dots are representing the zerosudirsen. The gray line that you barely see is the natural history data that's published. So here we're looking at lung function with zerosudirsen compared to published natural history data. And you're looking at 24 months, you can see that there is improvement in lung function versus if you look at the trajectory in natural history. We know that cardiac and pulmonary function is very important in DMD. So this is looking at a cardiac MRI and looking at left ventricular ejection fraction, or LVEF, with zerosudirsen. Again, this is looking at a 24-month period, and you can see that there was improvement for the LVEF, and this is 24 months. And the mean age at baseline for these individuals was about 12 years of age. So I already shared the 6-month data, but we also released this data most recently at MDA. So we were very grateful to get these optional biopsies from 4 individuals that were on the 20 milligrams per kilogram every 4 weeks for at least greater than 12 months. And you can see the unadjusted and adjusted value of dystrophin that is shown in the dark blue line. So as far as next step, we have completed the registrational expansion cohort. Well, I just shared with you the positive top-line results for the registrational expansion cohort, and we are planning for accelerated approval in US in Q2 2026, and we're hoping to launch in Q1 2027. So this will be our first launch. We also just most recently announced our phase 3 confirmatory study that is called Forzetto. So we're excited to move this program forward. And with that, I'd like to thank all of you and all the families and patients that participated in the study and all the clinical sites. Without you and all the clinicians, without you guys, we would not be here. So thank you. And thank you for your attention.
Let's hear it for Lena. I know it's always so hard when you have technical problems during your presentation, but you handled it great. Next up, we have Christine from Entrada Therapeutics to talk about their endosomal escape platform.
Hello. I'd like to thank Cure Duchenne for what they do every day for the community, but also for having us here. This is a pretty exciting time for Entrada. We're really happy to be on stage with some new, very early data. And so I'm going to walk you through that data briefly, but also describe our pipeline and our program a little bit. Here's the disclaimer slide. It is important to point out this is an investigational medication that we're talking about, and I'm also talking about really early data. And so, there's some forward-looking thinking and statements, but we're really trying to be good partners to the community and share the data as early as possible. And so, thank you for coming on this journey with us. So we really try at Entrada to make patient commitment not just a tagline. We really want groups like Cure Duchenne to hold us accountable and to help us do better every day in supporting the patients. We feel like we've gotten a lot of input from patients on our protocols, what it's like to be a patient in a clinical study. We know there's a big onus. And we really try to educate and bring awareness to not just our studies, but the disease in general. Entrada really started with a simple mission, which is to provide new therapies in drugs that deliver intracellularly. So we have a few faces here you might recognize: JJ, Max, and Franklin, and we're really inspired by what they do and how they persevere, and it's really important to us to model that same behavior. So Entrada may be new to you, but it's been around since 2016. We're headquartered in Boston. We have about 150 employees, and most of us are scientists. We have drug development labs, we do our own drug design, and we're really rooted in the technology, not just to deliver drugs intracellularly, but to understand Duchenne as a disease. We're a top place to work, which is really nice when you want to come into work every day and you feel inspired by what you do. So instead of explaining the technology to you, I'll let this video do it.
If you were to look inside the cells in your body, you'd see a flurry of activity. Every part plays an essential role in keeping the cell and the body healthy. With all these critical functions taking place, it's no surprise that roughly 75% of all disease-causing targets are located inside cells, yet most are still considered inaccessible. There are two major challenges to delivering a therapy to a target within the cell. First, getting the therapy inside the cell. Then, most importantly, avoiding the cell's natural clearing systems, so the therapy can successfully reach its intended intracellular target. Some small molecules can permeate cell membranes, but then tend to be rapidly cleared by the body before they reach the intended tissue. They also may fail to bind effectively to their intended target and can be associated with off-target effects. On the other hand, biological therapeutics like oligonucleotides, enzymes, and antibodies are generally potent and specific, but are limited in their ability to reach their intracellular targets. As a result, high doses are often required to produce a therapeutic effect, which can cause toxicity. Even when a biological therapeutic is successfully internalized, only about 1 to 2% will escape from the endosome to reach its intended target. At Entrada Therapeutics, we have developed a proprietary library of endosomal escape vehicles, or EEVs, with the potential to enable efficient intracellular delivery of highly specific and potent therapies to disease-causing targets previously considered inaccessible. EEV therapeutics consist of two essential parts: an EEV and a therapeutic modality. An EEV is a small peptide from our proprietary library. Its cyclic structure ensures rigidity and stability. The EEV is chemically linked to a specific and potent therapeutic modality, such as an oligonucleotide, enzyme, or antibody. Once inside the endosome, the low pH enables the EEV therapeutic to rapidly and efficiently exit the endosome and enter into the cell's cytosol to access its intracellular target. Unlike biological therapeutics, approximately half of an administered EEV therapeutic can reach its intracellular target, At Entrada Therapeutics, we are driven to transform the lives of patients by establishing EEV therapeutics as a new class of medicines.
So what you heard was a different way to deliver a drug like an exon skipper. And we have multiple programs ongoing right now. What I will really focus on is our most mature program, which is our program for patients amenable to exon 44 skipping. But we also have an active program in exon 45 skipping, and we're doing the groundwork for our program in exon 50 and exon 51 skipping. We also have licensed one of our drugs to Vertex for another neuromuscular disease, DM1 or myotonic dystrophy 1, and that data will be presented sometime this year as well. So where are we in our research? As you all know, there's a variety of things that have to happen before you get into actual participants who have Duchenne's. So we've completed animal and cell data. We've done study in healthy, normal volunteers. And where we are now is the early stage of our clinical trial program in our 44 exon skipping program. We call it cohort 1. It's 8 participants at the lowest dose that the protocol is allowing. So here is a schematic, probably pretty familiar to you all by now, seeing all this great clinical trial data. But patients were age 4 to 20. They had to be ambulatory, and they had to be amenable for exon 44 skipping and on stable steroids. What we're talking about right now is the first cohort. That's 8 patients, 6 of which were on the drug at 6 milligrams per kilogram, and 2 were on placebo. After they are treated for 3 doses, and the doses are 6 weeks apart by infusion. And at the end of the study, after 3 doses, the muscle biopsy is performed. And so the data I'll show you is 6 weeks after their 3rd dose. They then can go into an extension or an open label period and a follow-up period. We also have cohort 2 actively being treated right now, which is the 12 milligrams per kilogram cohort. We also have the 45 program ongoing, and we've completed dosing of those 8 participants at the lowest dose, and we'll have data out on that later this year. So who are the patients? When you first look at this, you look at the age, and the average age of the placebo group was 13.5, and the average age of the treated group was 9.3. It may seem like a big difference between the placebo and the treated group, but you know, the oldest patient in the trial was in the treated group. These ages might seem old to you for an ambulatory study, but those of you who are familiar with exon 44 amenable mutations, those patients tend to be a bit more healthy in terms of they ambulate till much later in life, and 17 to 20 years is about the average age that they may lose ambulation. The other thing to notice about this study is as they tend to be healthier, the exon 44 subjects tend to have higher baseline dystrophin. We had a lower level of dystrophin at baseline than has seen in other studies, like Avidity's recent study. There was an average of 7% dystrophin at baseline. So the most important thing to get out of the study is safety. It's really a dose-finding study and a study to understand the toxicity of our drug. So the drug was quite safe. Headache was the most common adverse event, and all adverse events were mild to moderate, and they resolved on their own. There were no low magnesium levels seen, which is really important when you're looking at this kind of oligonucleotide exon skipping therapy. It's the renal safety findings that you're most interested in. And so if you look at these renal biomarkers, you can see in green here, compared to in orange, the placebo, really there was no kidney safety issues. The placebo group and the treated group were all within each other. So now we get to exon skipping and the dystrophin. And so exon skipping really is measuring the amount of exon skipping that's happening after treatment. There's a base level of exon skipping that's obviously going on. That's why these kids have, a baseline 4% dystrophin. But what we're looking for is what is the dystrophin production, what is the exon skipping that's happening over and above that. And then we're also looking at, dystrophin as well, normalized to the muscle. And so what we see, to be quite frank, is a little bit less than we were hoping for in terms of exon skipping and dystrophin percentage. See about a 2.3% increase in exon skipping and similar 2.3% in dystrophin. Now remember, this is 6 weeks after the last dose. The oligo has to stay in the cell that long and continue to do this action. But based on our modeling, et cetera, we really are excited for the next dose, which is double the dose, which should give us an exposure that we feel will increase this dystrophin production. What we didn't really expect to see in 4.5 months' time was an effect on function. The study was not powered to look at function, but it's something that we looked at, and it's not easy in a phase 1/2 study to show data that doesn't unblind the data, right? These participants are still being actively treated in the open label period, and then they will go into a long-term extension. And so it's really important that we don't unblind the clinicians, we don't unblind the patients. So the data we can show you is a little bit limited, but what we're going to show you is the time to rise velocity, which you all have been well versed in over the last couple days. It's a really good measure of function. And the way we're showing this is a little bit complicated, and I apologize for that. But basically what we're looking at is the time to rise velocity versus the average from the treated group versus the placebo group. In the placebo group, both of the patients lost function, but in the treated group, what we saw is the majority of patients in time to rise, and hence time to rise velocity, got better. And got better, significantly better compared to placebo by a statistical test, but also better by a measure called MCID, which is a meaningful clinically important difference, or the minimal clinically important difference. And so we see about 3.5 times that MCID in 4.5 months of treatment. It's early days, and it certainly gives us something to look for in the next data readouts we have in this program and the other programs. But one of the questions that comes from this kind of data is, with a low dystrophin level, how are you getting a functional change? And our technology gets into cells, gets into muscle cells, cardiac muscle cells, but it also gets into satellite cells. And we've shown that through the last couple of years through preclinical animal models and in vitro studies. And so we feel that this early penetration of satellite cells may be causing a functional change earlier than we had expected, but it's certainly something promising and something we're going to be looking for in our next data readouts. So the summary is the treatment was well tolerated, there were no serious adverse events, and no one left the study due to treatment emergent adverse events. The kidney health markers all stayed within range, which is great. And then we saw a difference in functional benefit that we really weren't expecting. And so we think this is aligned, as I said, with our mechanism of action, and we're doing some more experiments to shore that up. So where are we in our other programs? As I said before, the 45 skipping program is ongoing, and we've dosed that first cohort, and we will hopefully be dosing our second cohort really soon with a higher dose. And we will be presenting that 45 cohort data at WMS in Hiroshima. We have an Elevate 50 and 51 program. Those are going through the process. We've had a lot of the preclinical and the tox work done, and so we're just finalizing the protocols based on the data that we're getting from the other two studies. So many people to thank, but let's thank again Cure Duchenne for holding us accountable and helping us really be a part of this community. Thank you to the patients and to the families. We really do understand that clinical trials can be positive, but they're also a lot of work. And we really, really appreciate the participants and their families. And we also really owe a debt of gratitude to our very committed clinical trial sites, the PIs, the staff, et cetera. And then I encourage you to stay up to date. You can sign up for our community updates and, and go to our website and learn about all the recent data that's coming out. Thank you again, and it's really wonderful to be here.
Thank you, Christine. So next up, we're lucky to hear from Lorna Hill from Wave Life Sciences, who has a little bit different chemistry for how they're targeting.
Hi everyone, good afternoon, and a huge thank you to Deborah and the team at Cure Duchenne for inviting Wave Life Sciences to present an update on our Exon 53 skipping drug, investigational drug, which is in the clinic just now. So I'd like to introduce myself. I'm Lorna. I'm the program lead for Duchenne at Wave. I joined the company back in November, and I've actually worked in Duchenne for the last 8 years. I continue to learn every single day from the community, from colleagues, from other companies, and it's amazing to see the science evolving. So a little bit about Wave. The company was founded in 2012 by two brilliant scientists, Dr. Verdeen and Dr. Wada, and they were pioneers in RNA therapeutics, and they were passionate about driving the discovery and development of RNA therapeutics that were going to be life-changing for patients. Interestingly, the name Wave comes from a combination of their surnames, so W.A. from WADA and V.E. from Dr. Verdeen. So that's how the name came about. As I've said, this is an investigational drug, and we'll continue to keep updating the community as new data evolves. So a little bit about the importance of dystrophin, which we all understand here, and the difference between muscle that's not affected by Duchenne and muscle that is affected by Duchenne. And we know that the stem cells and muscle regeneration is impaired in Duchenne. And while stem cells are activated following inflammation and injury, these cells cannot replenish, and fat and scar tissue forms in its place. And you can see the difference here between the cross-section of healthy fibers and DMD fibers. So this is the goal of exon skipping drugs, is to restore dystrophin and strengthen the muscle again. So Wave N531 is a phase 2, is in a phase 2 open label forward 53 study evaluating safety, dystrophin expression, muscle health, and functional outcomes as well. So it's a 3-part study. Part A evaluated different doses and a dosing regimen of every other week. 3 patients in that study. And then Part B is a 48-week study looking at 10 milligrams per kilogram every 2 weeks in 11 patients. These 11 patients moved over to an extension study, and following new data, we discovered we were able to reduce the dosing regimen burden for patients and look at a monthly dosing regimen. All patients have the opportunity to expand into an open label extension, and all of those patients are now in that, have chosen to stay in that open label extension study. There is a part C of the study. It's 24 weeks. These patients can also move into open label extension. There's 13 patients in that study, and the dosing remains at every 4 weeks. So these are the key things that we're looking at. Is the drug safe? Is it getting to where it needs to be? And is it doing what it's supposed to do? So what we are observing is a favourable safety and tolerability profile, no SAEs. And the muscle is getting to where it's supposed to, into the stem cells, into the muscle fibers at high muscle concentrations. We're observing high exon skipping, consistent dystrophin expression, and we're seeing significant reductions in muscle fibrosis and creatine kinase, which is really driven by a decrease in inflammation and scarring, and this important transition from regenerative to mature muscle. We are seeing functional benefits as well, which we'll go into. Patients in the study were aged between 5 and 11 years old, and all patients were stable on steroids. And these are the, you can see a list here of the functional baseline values. So looking at safety and tolerability, we're not observing any serious adverse events, and all drug-related treatment emergent adverse events were mild to moderate. You'll see that there was one TEAE, so this is a treatment emergent adverse event that was described as severe. This was a patient who had gastroenteritis. This was self-limiting. The patient spontaneously recovered and was doing very well shortly after. We have not seen anything other than this of note. So here we're going into the dystrophin results, the exon skipping, and you can see on the left-hand side here, we're looking at the mean dystrophin expression and exon skipping over 24 and 48 weeks. So we're seeing 54% exon skipping, which is showing that the investigational medicine is getting into the nuclei of the muscle cell, so it's able to produce dystrophin protein. And we're seeing mean dystrophin expression of 7.8%. Just under that table on the left-hand side, you can see that there's a 61-day half-life of the drug, which supports monthly dosing. So this is less burdensome on the patient coming into the clinic once a month for dosing. And also it means that patients don't require an indwelling port to administer the intravenous infusion. On the right-hand side here, this shows the consistency across boys in the study, and we saw that 88% of the boys achieved greater than 5% dystrophin expression. Here we're looking at creatine kinase as compared to baseline on the right-hand side, and we saw a 50% reduction in CK from baseline to week 48. On the left-hand side, when we looked at the muscle, we could see a decrease in muscle fibrosis between these two time points. And we could also see that when we look at the individual patients, every single patient, we observed a decrease in muscle fibrosis. Here you can see the muscle biopsy samples, and if we look across the ways, this is what patient 1, patient 2, we observed the same findings, really very similar findings across all study participants, in that where you see the red picked up by the stain, this is reduced scarring observed over time, and you can see improved organization and uniformity of the muscle cells. So if we look at functional outcomes here, we're looking at the time to rise. So we compared, you can see that on the blue line on the chart there, that is N531, and above that you can see the natural history control. Typically when we look at a slide like this, we think going up is better, but actually this is taking longer to get off the floor. So we're seeing that the trajectory from the treatment is lower, so that the patients are slowing their rate of decline with disease, and a 3.8 second improvement with WAVE N531. We also saw positive trends with NSAA and in muscle grip strength as well. So we think we have a differentiated profile here that addresses key unmet needs for boys with Duchenne who are amenable to exon 53 skipping. And we are interested in developing other exons in a preclinical setting right now as well. So in summary, we see favourable safety and tolerability, and we see that the investigational drug is able to reach where it needs to, including the muscle stem cells. We're seeing high consistent exon skipping and consistent dystrophin expression and reduction in muscle fibrosis, inflammation and scarring. and we see favourable evidence of functional outcome benefit. And in our preclinical studies, we also observed the ability of N531 to reach heart tissue and the diaphragm, importantly, as well. And I think this dosing frequency of monthly is an advantage for patients who are on chronic therapies in the case this is approved. So next steps for WAVE, we are really excited that we are progressing our study, and the last patients will transition from Part C into the open label extension, and we plan to submit a new drug application this year to support accelerated approval of N531 with monthly dosing. And we just want to recognise all the effort it takes from families and patients and clinical trial sites to invest time and energy into clinical trials. It's not it's a huge undertaking. We recognise this at WAVE and want to thank everyone for their participation. And just want to thank all of you here. I've had some amazing conversations this week. It's been a real pleasure to meet with you all, patients, families, Cure Duchenne, and peers in other companies as well. Thank you.
Thank you, Lorna. It was great to have that update. So now for the grand finale, we have Matt Fisher from BioMarin. He's been waiting patiently backstage, so let's give him a warm welcome.
Hello, good morning, good afternoon. My name is Matt Fisher. I'm an Associate Medical Director at BioMarin and the Medical Monitor for our Duchenne muscular dystrophy study. It's called BMN 351. So this is just a forward-looking statement. And today I'm going to be discussing what BMN 351 is, some of the results we've seen in our phase 1/2 study, and some of the next steps for the program. This is just an overview of DMD, but I think I could probably skip this slide out. So BMN 351 or nivodersen is a second-generation ASO. So we at BioMarin had developed drisapersen, which was our first generation, and this is kind of building on the things that we'd learned from that. So BMN 351 targets a new splice regulating site in a dystrophin gene called Exon 51. It's being researched to see if it's able to get into cells and work inside them. And it's also designed to lower the chance of unwanted immune system responses against the medicine and to last longer in the body. So, because this is a phase 1/2 study, our main kind of goal or primary endpoint is looking at safety and tolerability. But the secondary goals we're looking at is pharmacokinetics and pharmacodynamics. So really what that is how the body processes the drug and also the amount of the drug that is getting to the muscles. We're also looking at, dystrophin expression and the ability the drug has to improve functional outcomes for boys with DMD. So this is just an overview of the study. We've actually recruited now all 18 boys into the trial. It's divided into 3 parts, so group 1, 2, and 3. Group 1 is the lowest dose at 6 mg per kg. All of these boys have now been dose escalated to the group 2, so at 9 milligrams per kilogram. And we also have a third group, which is at the highest dose for 12 mg per kg. And as of February, we've had 68 weeks of the low dose BMN 351, 43 weeks of medium dose, and 10 weeks of the high dose, but I guess add like 3 or 4 months. So this is just an overview of the study and its kind of totality. So part 1 was looking at dose escalation. So we started at a low dose to ensure that the drug was kind of relatively well tolerated. It started at 0.6 milligrams, and over 8 weeks this progressed to 6. As I've mentioned, these boys have now moved to 9 in the 6 milligram dose. And then we are now moving, so from our group 1 participants who've been on the trial the longest, we're now moving into the extension study, so that's 351-202. This is just an overview of some of the safety findings. So the most common AE related to the drug is changes in kidney markers, so cystatin C, which is kind of a measure of an indirect measure of kidney function. So 64% of the boys have had an increase in that. Also changes in liver markers, so GLDH, that's 29%. I'm the medical monitor, so throwing up, runny nose, cough, diarrhea, common cold it's perhaps it's not a persistent problem. Maybe you see it once or twice. No participants have had their dose of BMN 351 lowered due to an AE. Fortunately, no one has died due to an AE, and there have been no severe AEs related to the study drug. This is an overview of the dystrophin measurements. So we're using LCMS to kind of come up with these results. At 13 weeks, the boys in the 6 milligram dose had a 2.2% increase in dystrophin expression. And at 25 weeks, it was at 2.4%. And then the higher dose, so at 9 milligrams per kilogram, at 25 weeks, there was a 5% increase in dystrophin expression. I think this is quite an interesting slide. So this is looking at kind of creatine kinase and lactate dehydrogenase. So creatine kinase is released when muscle is injured. So if you have DMD, your muscle is being injured. So typically these boys have very high creatine kinase levels. So if you can see the gray line there, that's a placebo, but it's a historical placebo from our first trial in dry person. And as you can see, the creatine kinase remains unchanged throughout. But then if you look at the two lines below that, the blue and the purple, at the kind of low and medium dose, you can see about a 10,000 reduction in creatine kinase. And I say just from looking roughly, boys normally start at maybe with a 20 to 15,000 level of CK. So it represents quite a significant reduction. And noticing similar patterns as well with lactate dehydrogenase. And then this is just looking, so the muscle test data. So this slide here, this is like the functional data, so it's how the boys are performing. Using the NSAA, which is a pretty standard measure for measuring functional outcomes in DMD. You can see there with the 6 milligram dose in the purple, the dashed line is the historical placebo, and you're noticing there kind of typical trends of a reduction in the NSAA over time. But what we have noticed at 49 weeks, sorry, that's the medium dose, not the 6 milligram dose. What we've noticed at 49 weeks is that the boys have basically maintained the NSAA score and have even had a kind of a slight improvement. And at the 6 milligram dose, so this is obviously, they've been on the study longer, so 73 weeks, we are continuing to see kind of no change in NSAA and actually a slight improvement. There are no questions. And finally, I just want to say thank you particularly to the boys and their families for taking part in the study. It's a big commitment, and we appreciate it. Thanks very much. Thank you.
Thank you so much, Matt. That's our session for you. We're now delayed for lunch, but I hope you enjoyed everything and look forward to talking with you guys later. Everybody will be available if you'd like to follow up with them directly. Thanks.